So welcome to this class. In the last class, we have just briefly gone through introduced the reconstruction of the needle specimen. We briefly described about the point projection model and how this model can be used in a reverse point projection model, which is used to reconstruct the actual needle specimen after the field evaporation. Okay, so as in the last class I mentioned, the reconstruction is having the two important aspects. One is the lateral positions, another one is the depth coordinate. Okay, so fundamentally the first approximation is that only the atoms that are protruding most from the surface are field evaporated. Okay, so which is subjected to a strong electric field, okay, locally at the atomic scale. So what the atoms which are protruding most near as compared to the neighboring atoms, those gets field evaporated. Okay, this particular assumption, okay, underpins the shell model. Shell model which was proposed by Murray in 1967. Okay, so here the atoms actually they are likely to evaporate. They are likely to evaporate which are actually belongs to a thin shell at the surface. Okay, so if this is a shell, then there should be a thickness of the shell, which actually directly relates to the penetration of the electric field into the surface of specimen. Okay, so the atoms which are protruding at the surface, they get field evaporated, and it was assumed that it belongs to a thin shell. Okay, and that thickness of the shell is directly related to the penetration of the electric field into the surface specimen. Okay, and this penetration depth, okay, is of the order of is much less than the d-spacing of that particular crystal structure. Okay, this thickness is corresponds to the skin depth. Now this electrostatic electric field F, okay, so we know that the electrostatic electric field is proportional to the surface charge density, which is sigma E divided by epsilon naught. Okay, this is the field, electrostatic field. And E0 is the dielectric permittivity. Okay, and surface charge density is nothing but the number of charges in a thin shell with a thickness of dp. Okay, so the dp corresponds to the penetration of electric field, the distance of penetration of electric field into the surface of the specimen. Okay, so here then the dp is given by Ne divided by F E naught. where the Ne corresponds to the electron density of the volume. Okay, and the penetration depth can be calculated, is calculated to be in the range of few picometers, which is less than the interplanar spacing of the crystal structure of the specimen. Okay, so this shows that theoretically the atoms gets field evaporated by layer or atom by atom, layer by layer, from the outermost shell, towards inside. Okay, so the sequence, so here the important aspect is the sequence. The sequence in which the ions are detected, this sequence relates to the depth of atoms which are removed from the sample surface. So this sequence is very important to understand the depth at which these atoms are removed from the original sample. Okay, so most of the protocols rely on the exploitation of this particular order or the sequence of the detected ions. That's how the Z depth is created. So we'll just go through some of the equations to understand how this Z is calculated. Okay, so before going to the Z, we know that the detector coordinates will be given by xt and yd. Okay, so this enables the xy position of that particular ion. Okay, and with the study of the sequence, okay, the protocols for the sequence of the removal of atoms, that gives you our Z position. This is how we get the x, y and z position in 3 dimensions. Okay, and the mass to charge ratio gives the time of flight. Okay, so here each ion which is detected will have a atomic identity, and also the important point is the atomic volume. Okay, so this particular atomic volume is used to determine the Z or the depth of Z sequence of the removal of atom from the tip surface. Okay, so basically the process of reconstruction requires, the first important point is the knowledge of radius of curvature for each ion detected, evolution of radius of curvature of each ion detected in order to compute the original location at the specimen surface. Okay, so you know that the Shank angle is not 0. Okay, the specimen gets, so as this the atoms get ionized and field evaporated, the specimen gets blunted. Blunted means the radius of curvature or the radius increases or the curvature decreases. So to maintain, due to which there is a reduction in the radius of curvature, it means that the electric field also drops. Okay, and this induces drop in the detection rate. Okay, so to counterbalance this detection rate, if you want to keep at a constant detection rate, then the voltage is increased, high voltage is applied during the experiment to maintain a constant detection rate, because there is an evolution of radius or increase in the radius as the field evaporation progresses. Okay, if we assume that the electric field is homogeneous, across the specimen surface, and is this electric field is very close to the evaporation field of that particular material, then actually the radius of curvature you can get by the equation V by KFFE. Okay, assuming that if electric field is homogeneous and it is near to the field evaporation field of that particular material, you can get the instantaneous radius which is given by the voltage which is applied by the field factor Kf and the Fe, which can be estimated. Okay, so as we described, Kf is the field factor, okay, and Fe is the evaporation field, which is we are assuming that is a constant for a given material. Okay, and here the voltage So here the voltage can be given as the DC voltage which is applied plus the pulsed voltage in HV pulsing. Okay. Now we will talk about the depth coordinate. Z. Okay, and this Z is actually deduced from the ion evaporation sequence, just now we described about the importance of the sequence of atoms, correct? So this is deduced from the ion evaporation sequence. So as the ions are removed by atom, or we can tell as a layer by layer, the specimen surface becomes farther from the detector. Okay, the specimen surface becomes farther from the detector. For each ion added to the reconstruction, so during the emitting surface shifts by dz. Okay, and the z tip, which I plus 1 we can keep, is equals to z tip for the ith atom plus the dz. So this is the equation we use for determining for defining the z, and that evolves during the field evaporation where the surface shifts opposite to the detector opposite to the detector as the evaporation takes place. Okay, so the calculation of depth increment is the one term which is used to calculate the depth is the volume analyzed. Okay, so and this analyzed volume is the sum of volumes for each evaporated ions, which can be given by omega I. Okay, so volume evaporation can be given as number of ions evaporated, their summation of volume, which is can be equals to what we can it as n EVF divided by this one. Okay, so here omega corresponds to the average atomic volume and NEVAP corresponds to the total number of evaporated ions. Okay, so we know that not all the evaporated ions will be detected, so there will be a detector efficiency. So we can write it as a ND is equals to ND is the total number of detected ions and N is the detection efficiency. Okay, fine. So this is the ND equal to eta into NEVF. So the analyzed volume can be related to the geometry of the specimen. Okay, so here okay, so the analyzed volume can be related to the geometry of the specimen. So here we can give as a so V EVAP is equals to 0 to Z max integrating WV Z dz. Here this particular W dz is a function with that describing the increase in the analyzed volume. Increase in the analyzed volume of the specimen as the specimen is field evaporated. Okay, so the dz can be given as the atomic volume divided by NWVZ. Okay, so most of the protocols depends upon the evaluation of this WVZ. Okay, which is the function of function describing the increase in the analyzed volume or the specimen as the specimen is field evaporated. Okay, so this is the basic thing which is related to how to evaluate the Z, and combining this point projection model with this, which is proposed by Bass et.al, a simplified point projection model. Okay, so we can, as magnification of projection is equals to d by d, which is equals to which can be written as L by image compression factor divided by R. Okay, so here the d is the distance from the ion impact point to the center of the detector. Okay, and the d is the distance between the specimen axis of the projection of the ion into the plane. So in this figure it is written, it can be understood. Okay, so here the M projection is equals to d by d, which is equals to L by R. Okay, so d is the distance from the ion impact point. This is the ion impact point to the center of the detector, and the d is the distance between the specimen axis and the projection of ion into the plane. This particular into the plane which is perpendicular to the specimen axis. Projection of that particular ion into the space which is perpendicular to the projection axis. Okay, so assuming that ions are projected along straight trajectories as we described for the reverse projection model, so the relationship between x and y position towards xd and yd can be written as x is equals to xd divided by m projection, and y equal to yd divided by m projection in a 2-dimension. Correct, and the field evaporation takes place about a volume. Correct, in this figure you can see that this is your needle, okay, and the field evaporation takes place for a volume. Correct, and this field evaporation V app is can be written as SA into dA. Okay, so here the SA is the analyzed area, 2-dimensional area, analyzed 2-dimensional area, which is, okay, this is SA, and the dA is the analyzed depth. Is the analyzed depth, which in the schematically it is written as dA. Okay, so the size of the region of specimen surface lying within the field of view is called the SA. Within the field of view, the size of the specimen in 2-dimension is represented as SA, and we can tell that volume of evaporation can be assumed to be SA into dA. By small angle approximation, that this SA can be assumed to be the reverse projection of this particular SD, which is projected on the fluorescent screen, on the screen. Correct, so reverse projection of the detector in the plane tangent to the specimen axis can be written as SA, can be written as SD divided by magnification of projection square, as this is an area. Correct, so this is written. So SD is the surface of the detector, SD is the surface of the detector, and M projection is the magnification of the projection. Okay, so assuming with these equations, assuming the atomic volume is distributed homogeneously across the area, okay, atomic volume, then we can write W V Z is equals to SA, and from the earlier formula R is equals to V by KF FE, evaporation field, dz is written as the atomic volume divided by efficiency, and this particular function which is responsible for the projection, and M projection can be estimated by L by image compression vector R, and SA is equals to SD divided by M projection square. By using these 4 equations, these 5 equations, you can write it as SA is equals to SD divided by L divided by xi R whole square, which can be written as dz equal to atomic volume L square divided by SD xi R square, and dz again can be written by using this equation. We can write as atomic volume L square KF square field evaporation field square divided by efficiency SD xi square voltage applied. Okay, so this projection generates the atomic positions within the plane tangent to the specimen axis. So in this figure, as I told you that if you see this particular figure, okay, so you are assuming that the atom which is present on the tip surface is projected onto a plane which is perpendicular to the specimen axis. Okay, so we need to correct this factor because it is a projection, correct? So we have calculated now dz. Now it has to be added another correction which takes care of this plane tangent to the specimen axis. Okay, so this can be deduced by at positions, okay, by for the ith ion, the dz dash, okay, can be computed as ri 1 minus xi plus y1 square divided by ri square. Okay, so for each ion we can calculate this dz dash, which is a correction which has to be included in the depth, which is calculated by this particular formula. Okay, so the z position of an ith ion can be written as zi is equals to summation of I dz plus dz I dash. Okay, so this is how the z coordinate is calculated based on the assumption that the field evaporation is taking place atom by atom or the layer by layer method. Okay, so in summary, what we got to know is to build a reconstruction, we need 4 basic parameters which has to be calculated. The first one is the image compression factor. The second one is the field factor. The third is the evaporation field. And the fourth one is the detection efficiency. N. Okay, and for Z, as I told you, we are assuming that the fields, the atoms are evaporated by layer method, and for calculation of this, we are using the atomic volume. We are calculating the atomic volume, and from the atomic volume, we are actually calculating the depth, the Z of the particular ion. Okay, so I hope so in this class we are explaining about we have explained about the parameters which are needed to estimate the ion position on the detector, especially the x and y position, okay, and the z position, which is calculated by the atomic volume. Okay, so in the next class what we will go through is we will go each of the 4 reconstruction 4 basic parameters and how to calculate from the data obtained from the data obtained from the atom probe or the field ion microscope. Okay, so with this I will end this class today and we will meet into the next class. Thank you.